Okay, I will get going. We're missing a few, but I'll check again before we leave. So welcome back. I just saw you 2 days ago, and then again, we are meeting again on Friday. So we will meet Monday, Wednesday, Friday, just to make sure I see you regular and check in with you often enough. And so, a couple of things. Don't forget, you have a quiz tomorrow over your prefixes and suffixes. Remember, it's your second quiz. So it'll be all of section 2 plus any 10 words that came before it. So there'll be about 33 questions. These 20 new words plus 10 of the old words, and then your 3 where you tell me what the word means. So that way we're building a little as we go instead of doing it all at once. So don't forget the old words. Look back over them, because they're on your quiz too. So that quiz is tomorrow. You can do it anytime I talk to Miss Upmore, just to have some flexibility, because especially with the virtual world and your internet and Canvas and things being up and down, I'll have it due at midnight. So I'll open it at the time around noon when class starts, and then I'll close it around midnight. So you'll have plenty of time on that one. Do your daily attendance each day. Our web meetings are here, so if you miss something, again, you can do it. I will tell you that it does take me a little bit, because when I do the recording, it doesn't tell me that it's ready until like midnight or something, or 11 p.m., and I'm already in bed. So it does take me a little bit, probably the next day, to get that loaded, but it is there. So cells is where we are now. You did your plasma membrane stuff on Monday, right? We did your cell, the parts of the cell, all those organelles, and then plasma membrane, remember, is the same as your cell membrane. It's that outside barrier controlling what can come in and out of the cell, and you did this plasma membrane activity. If you read the directions, if you don't have a printer, totally fine. I know not everyone has a printer at home, so you were able to hand draw it if you want, not a problem. And then since we have this barrier for the outside, we need to let some stuff in and some come out. So now that's where cell transport comes in. And that's what you did yesterday, is your cell transport, right? Some things can come in on their own. Some require some energy, right? We don't like uneven concentrations of things. So stuff will diffuse till it gets even. So we talked, some things move on their own straight through that membrane. So if it moves on its own, it's passive transport, does not require energy, goes straight through. That's simple diffusion. If it uses a protein to help it, because maybe the molecule's too big to fit, okay, that's a facilitated diffusion. Osmosis is if we're moving water, doesn't require energy. And we're going to osmosis side of things. And then filtration is also passive, and this is what happens in your kidneys. On the other side, some things are active. They require energy to make them happen. So just like, I'm putting more and more and more and more, I'm going to have to do some energy and some work to make it all fit. So that's active transport, goes against that concentration gradient. And that's why it takes energy, that ATP, to make it happen. And so that's your solute pump that's like acting as a pump to push it over, and bulk transport, where I'm moving large amounts of things. Okay, and then you did some practice, and I want to talk about this practice because I've had a couple of questions. So when you open it, you make a copy, and there's a website, a Google Doc, where you go to this website, play with the molecules. You can see the molecules moving across the membrane, different types of molecules. So you will answer the questions. And remember right here at the top to put it in your anatomy folder that's shared with me. And then answer the questions, but down here, you're just going to put a check box to all that apply. So for oxygen, is it moving from high to low or low to high? And then is it simple, facilitated, a solute pump, or exocytosis, right? So you'll just put a checkbox in one of these and a check mark in one of these. Carbon dioxide, put a check here, put a check in one of these. Some of them might have more than one check. I can tell you water will have more than one checkbox over here because it does it simple diffusion, goes straight through because it has enough energy. And then also those aqua pores, so it has some help as well. So you're just putting checkboxes where they belong. And that was it from yesterday. And then today we're going to look a little more in depth at the water movement, and that's what causes what we call tonicity, is tonic solutions. And I talked about it in your notes, right? ISO means same, so this is where I have the same concentration. Stuff still moves, but it moves equally. So we're already pretty good and happy. Hypertonic, remember, water always goes to where there's more solute, so water moves outward. Because my cell lost some water, it starts to shrivel up. And then on the flip side, you have hypotonic, where I have a higher concentration inside, so water moves inside. I really like this video from the Amoeba Sisters. I think it explains it really well with a little cartoon. But they only really do isotonic and hypertonic, so that's why I threw in this video as well, because it does do all three in hypotonic. If you're interested, there's a video here which is really interesting by Chubby Emu, and they just do life. It's from a doctor. They do life application, and there's a story about a woman who drank a liter of soy sauce in 2 hours because she thought it was part of a colon cleanse, and this is what happened to her brain. Okay, this is real life. This is why we don't drink soy sauce in 2 hours. This is why you can get water poisoning if you drink too much water too fast. I mean, water's good for us, but too much too fast can cause a problem with our cells because of these tonic solutions. So this is just kind of a fun thing to watch if you're interested. I think it's really interesting. So, and then you do have a virtual lab that you're doing where you follow through the instructions and then you'll answer the questions for it. Tomorrow, because again, I won't see you tomorrow, so I'm going to go ahead and show it to you. Remember, you have your quiz. I will open it up and send you the access password. We're starting body tissues, and this you have not done before, so I really need you to pay attention. The notes are there as a Google Doc, so you can fill them in the PowerPoint. Just go through and fill in the notes as you go. I'll make some videos to break it into epithelial, connective, and then muscle and nervous. And you have a worksheet and then some review to do. This microscope I'm going to take off because that's for my in-person class because we have a lab we're doing with microscopes next week. So I'm going to come up with a new version of the lab activity because I don't want you to miss out on anything. So I want you to see these tissues because, I mean, it's really kind of exciting. They look very different. Your bone tissue does different things than your blood tissue because blood is a tissue. It's different cells working together. So blood is technically a tissue versus your skin. Like, they're all built differently. They look kind of cool. They look different under the microscope. So I'm going to come up with a new activity for us to actually do those. So just so you know what's coming next week as well. So, but this gets us through this week. And I will, again, I'll see you on Friday, so it's not a big deal. We'll talk about that then. But just to give you a heads up, so for tomorrow. But that's about it. If you have questions on anything, let me know, and I'll stick